So how many of you have heard of PET imaging? Good, a fair number. Okay, and probably you've heard of it in the context of medicine, biomedical research, but we can also use the same tools for plant research, which is great because I'm a plant biologist. So where is the best place in the world to develop or adapt these tools for plant biology? Brookhaven, obviously. There are only a few other groups in the world that apply this technology to plants. Brookhaven is the only one that brings together all of the tools, all of the expertise to look at the movement and distribution of different molecules in plants. So what I do here under the Department of Energy mission is I do basic plant biology research for bioenergy. So I'm going to start by giving some introduction to the challenges we're facing with energy right now, the societal and scientific challenges. I'll give you some background in the PET imaging tools and radio tracers, and then I'll talk about some applications in plant biology, focusing particularly on plant sugars, their movement and metabolism. So energy, it's central importance to our society, from war to the economy and even to the food we eat. We're dependent on energy-intensive agriculture to feed our population. The scale of energy usage is enormous, so the scale of the problem is also going to be enormous. We use, just in the US, the equivalent of about 850 gallons of gasoline per year. And most of that, 85%, is supplied by fossil fuels, digging hydrocarbons out of the ground, burning them. And this is basically a one-way street. We're burning those materials. Carbon dioxide is a byproduct, and that's released into the atmosphere. And we've seen over the last 100 years that we have increased the atmospheric concentrations of carbon dioxide. There are potential consequences of that. And also the other issue with this one-way street, the supply of fossil fuel is finite. Whereas renewable energy supplies about 6.8% annually. So what we'd like to do is increase the fraction of our energy supply that is renewable. One example, which I've already mentioned, is bioenergy. Bioenergy is basically growing plants for biomass, either burning that biomass or converting it to a higher value liquid fuel and then burning it. Now, this also releases carbon dioxide, but the process of growing the plants, producing the biomass, relies on photosynthesis, which takes carbon dioxide out of the air and incorporates that carbon into its own tissues. So this is more of a cycle. It's more carbon neutral, and because it's a cycle, it's renewable. So we could potentially have a long-lasting energy supply from bioenergy, as long as the sun keeps shining, which we all expect. It takes energy to make energy. And what this chart is showing, it's the fossil energy ratio, shows how much energy is delivered to the consumer per unit of fossil energy used. So as an example, gasoline, when you put 0.8 gallon of gasoline into your gas tank, it took a gallon of gasoline spent just to get it there. So you're really paying for 1.8 gallons. So this is inefficient. Most of the current systems are fairly inefficient. You may have heard of corn ethanol. That's a little bit better. But what we want to do is improve our efficiency. And this is, again, going to affect carbon dioxide emissions, atmospheric carbon. And also, it's going to make bioenergy more economically sustainable. So we're not going to need incentives and mandates in order to make this happen. We need research to make it happen. So what is biomass? I've talked about bioenergy. It involves biomass. So far, we've used cornstarch and sugar from sugarcane quite a lot. That's not the most effective approach. And the problem with that is that we're taking a product out of the food market And when we do that, we're going to increase the price of food. For you and I, that may not be a big deal, but for the people on the margins, we're going to be pushing people into starvation. And we don't want that. We don't want to make people's lives worse. We want to make people's lives better. So we can also use the inedible biomass, the fibrous or woody parts. You've probably heard of cellulosic biomass or lignocellulosic biomass, and that can be inedible parts of edible plants, like the corn stalk from the corn plant. It could be dedicated bioenergy crops like poplar or bioenergy grasses like miscanthus, switchgrass, I think we've all heard of, thanks to Bush, and sorghum. Why biomass? Well, there's lots of it. There's 56 billion tons of carbon dioxide fixed every year by plants through photosynthesis, 170 billion tons of standing plant biomass. And a study by the federal government, joint USDA-DOE study, suggests we could displace a significant amount of fossil fuel usage by producing 1 billion tons of plant biomass per year. So we're on about the right scale. And so let's look a little bit more closely at that biomass. 70% of all biomass, and now this is not just plants, but all biomass, is cell wall components. And if we could break out a cell, peel off the cell wall and look at what's in it, it's mostly cellulose and hemicellulose, which are made up of sugars. It's polymers of sugar. And those sugars can be fermented and turned into fuel. There's a substantial portion that's also lignin, which cannot be fermented, but the residue, after conversion of the other parts of the cell wall, the residue can be burned and used to energize this conversion process, the fuel production process. So what are the major challenges? One major challenge is taking that cell wall, converting it into the sugar subunits. It's going to involve use of microbes, enzymes to deconstruct the cell wall in a way that's energy efficient, that's cost efficient. And there can also be some improvement in the fermentation process itself. Glucose, we're good at fermenting. The 5-carbon sugars that tend to come out of hemicelluloses, not so much. But we're making progress, and I think we're going to be able to use those cell wall sugars. The other part of this is we need to develop dedicated bioenergy crops because we don't want to compete with the food crops, and also the characteristics that we're going to look for in a bioenergy crop are going to be a little bit different than what we want for a food crop. For example, we may be able to help deal with this conversion issue by making more of the plant produce sugars that can be fermented directly. And I'll come back and talk about that later. So another issue that we need to think about in producing these dedicated crops, and by the way, that's where my focus is as a plant biologist, is the providing information that can guide crop improvement. So one of those pieces of information, if you look, the red here on this map is indicating areas that are intensively farmed. It's actually a map of fertilizer use, but that's a pretty good indication of farming. There are some parts of the U.S. that we can't use. For example, we're not going to grow bioenergy crops out in the desert. It's just going to be too slow. We're not going to grow bioenergy crops out where I'm from in Maryland, near D.C. It's city, it's suburb. You don't have the land space. So what do? I think what's going to happen, and what the USDA and DOE are suggesting, is there's potential to use marginal lands. And these are lands that maybe they're near those prime farmlands that have just the right nutrients and water, but they're not quite good enough for food production. We've really pampered our food crops, and they need high nutrients, lots of water. So can we use other lands in order to avoid competition, not only at the level of the product, but also at the level of land use? And what can we learn from agriculture? We have a long history of agriculture. What we can see from the Green Revolution, which saved millions of people from starving over the last century, is that for food agriculture, we took plants that were large, selected for plants that were smaller, and that were putting more nutrients into the parts that we eat, usually the seed. On the other hand, for bioenergy, I just mentioned, we want to use the inedible parts as well. So what we really want to do for bioenergy is almost the opposite direction. We want to produce really big plants, as much biomass as possible. And maybe this is even at the expense of seed production. So what exactly do we want? I just mentioned fast growth. We want to be able to grow bioenergy crops on marginal land, so we want these crops to be stress tolerant in case those lands have low nutrient availability or low water. Maybe you need to be able to survive drought or high salinity. Another thing we can learn from agriculture is when you take a few types of plants and plant them out over a large land area, you're going to have pests evolving, insects, pathogens. So we need to make sure as we're developing these crops that we don't lose that hardiness, that resistance to pests, and the resistance to stresses. And finally, I touched on this before, the composition of the plant could be important as well. Things like high cellulose, which is made up of sugars, or carbohydrates, sugars themselves, would be beneficial. Things like low lignin, because lignin actually gets in the way of deconstructing those cell walls. And lipids can be used for biodiesel as well. So what do all of these things have in common? They're all dependent on resource allocation and chemical partitioning. And that's something that we can study really well with the PET technology here at Brookhaven. So we've selected grasses mainly now as our plant model. Rich mentioned that I worked a lot with poplar. So grasses have a lot of the traits that we want to see in bioenergy plants. This is going to look kind of like a repeat: fast growth, stress tolerance, so they can grow on those marginal lands, pest resistance. Another advantage, there are lots of transgenic and mutant lines available in the grasses. This is important because we need those as tools for research, not necessarily to deploy, but to understand what the plants are doing, we need those. And finally, there are lines with a favorable composition, for example, high sugar varieties, which I'll talk about later in the talk. So the challenge to me, as I see it, is how do we improve plant composition and at the same time have plants that grow fast and maintain that hardiness, maintain that robustness to stresses and to pests. So we really need knowledge as we go forward to make sure we're not losing those hardiness traits as we select for plants that are growing fast. And on the other hand, if we're looking specifically for traits that contribute to robustness, that we're not going to sacrifice growth in doing that. So that's my background on bioenergy, and now I'm going to talk about the tools that we use, the PET imaging, the radio tracers. Probably many of you have heard of isotopes before, but just quickly, in case any of you haven't, isotopes are variants of an element. They have the same number of electrons and protons as we see in this example of carbon. And so that makes them chemically similar. They're going to behave the same way in a chemical reaction. They're going to behave the same way inside of an organism. So that's very useful. But we can distinguish them by the number of neutrons, either by measuring the mass, or in the case of the unstable elements like carbon-14 and carbon-11, they emit radiation as they decay. And we can detect that. So isotopes have actually been used quite a bit historically in plant biology. And just a few high-level examples. One that you may have heard of, possibly. Melvin Calvin received one of those prizes that you've probably heard of, the Nobel Prize in Chemistry. And this was actually for his work figuring out the steps in the pathway of photosynthesis, converting carbon dioxide to sugars. And this involved heavy use of the isotope carbon-14. Martin Gibbs, unless you're a plant biologist, you probably haven't heard of him, but he actually worked at B&L. And he's considered a pioneer in the use of isotopes in studying plant carbohydrate metabolism. And there's an award, the Gibbs Medal, that's named after him, awarded by probably the top plant biology society, Association of American Society of Plant Biologists. So carbon-14 is great, and it was very useful, and it still has some uses. But there are some advantages to that other radioisotope of carbon-11, which is a positron-emitting isotope, and I'll talk about that more in a few minutes. One of the big differences here is that we have a higher sensitivity of detection, and because of that, the carbon-11 can be non-invasive, more so in some situations than carbon-14. So what I mean by that is if we want the same signal, the same amount of radioactivity, which we measure in curies, so if we want, say, a microcurie, we're talking about 10 to the negative 15th grams of carbon-11 versus 10 to the negative 6 grams of carbon-14. So this is 9-fold difference in mass, 1 billion times difference. So we can introduce much lower levels of carbon-11, and so we're not changing concentrations within the plant. We can look at it without perturbing it. That's what I mean by non-invasive. Another advantage, the half-life of positron-emitting isotopes like carbon-11 is on the order of minutes. And it's not just carbon. We have the other elements of life, carbon, nitrogen, oxygen. There are positron-emitting isotopes for all of those. So one of the advantages of that short half-life is that within a few hours or the next day, you can take the same plant and test it again. The radioactivity is gone. So we could look at the response of an individual plant to a treatment, let's say. Also, maybe a downside of that short half-life, you need specialized equipment to make it. How do we make it? How do we administer it? This is one of the cyclotrons that we use to make carbon-11. It basically accelerates a proton beam at high energy and blasts it into a target of nitrogen gas. The nitrogen gas is converted to carbon-11. And within that target, there's also a trace amount of oxygen. And because of that, what comes out of the cyclotron is carbon dioxide. Perfect, because plants use carbon dioxide for photosynthesis, so we have a really natural way to introduce that. So we can take the labeled carbon dioxide gas, send it to a plant within one of our shielded growth chambers. And how we actually do that is we'll clamp a leaf inside of a cuvette. We'll pass air over the leaf, and then for 30 seconds we'll introduce that carbon-11 carbon dioxide tracer, and this is the cuvette. And the leaf will actually take up quite a bit of the carbon-11, convert it to its own metabolites, and we can measure that fixation. If we take the leaf, we can look at what metabolites are labeled. And also some of those metabolites will be moved out of the leaf, and we can look down in the roots with, say, a PET camera at where that radioactivity moves to, where it's distributed to, not only as a snapshot, but how it's moving over time. And this particular prototype, a dedicated plant PET camera, is something that is being developed by Paul Vaska and Dave Schleier, along with a number of other people in physics and instrumentation division. So this means we can put it, it's small, we can put it into these controlled environment chambers where the plants like to be. One other thing we can look at, if we have radioactivity moving down through a leaf or a stem, we can use shielded detectors to see how long does it take to get to point A, detector 1, and how long does it take to get to point B, and we can use that to calculate the speed of movement just like the police do sometimes when they time you moving that fixed distance. Same concept. One of the other advantages of positron-emitting isotopes is that, I mentioned the PET camera. We can look dynamically at what's happening. And this requires the use of positron emission tomography. That's where the PET comes from. I don't actually pet my plants. Not often. So these isotopes emit a positron. When that positron collides with an electron, they both annihilate. All of the mass is converted into energy. The energy is released in opposite direction as gamma rays. Those gamma rays can actually travel through thick tissues like a skull. So we can we can detect the signal from, say, inside of a brain. So if you have two detectors, like I show here, in communication, we can figure out where in between those two detectors did that signal come from. And you can imagine if you have a ring of detectors and you start stacking rings, that you could reconstruct a three-dimensional image. Where is the radio tracer within a particular organism, whether it's a human looking at, say, a brain or a plant, for example, this sorghum stem. And just want to point out, this is one of the larger PET cameras, the human PET camera, where we can actually put larger plants in there and look at larger plants. That's where this image comes from. Yes, that bar is a half meter. So that gives you a sense of the technology. Now I'm going to talk about examples. And I'm going to focus on plant sugars, although we cover a lot of other areas. That's just due to the constraints of time. So Rich mentioned one of the first studies that was done here at Brookhaven with plants using positron-emitting isotopes was looking at plant responses to herbivory, to insect chewing. So I just want to use this as an example, a lead-in, so you can get a feel for what we can see, what we can do. And what I've done in this study is I measured a plant one day, I applied a treatment that night, and at the same time the following day, I retested, I remeasured that same plant. So we're looking at the response of individual plants to particular treatments. And you can see here some of where the gypsy moth caterpillar damage occurred. I caged some gypsy moth caterpillars on this tree. And in this case, instead of an actual insect, I used one of the plant's own signaling molecules. Plants produce jasmonic acid when herbivores start chewing, and that's one of their signals to respond, to start making toxins, deterrents. And we knew about that. So what's exciting is that after that first day, that treatment, we saw more of the carbon-11 introduced as CO2 to a leaf, leaving the leaf and going down to the roots, especially for the jasmonate treatment. And this led us to realize, maybe plants can actually take resources when they're under attack and bunker them underground where a leaf-chewing caterpillar can't get to them. And as Rich mentioned, this spawned a lot of research in response to this and some new ideas on how we can deal with insect pests. But what I really wanted to do is use that to lead in carbohydrate transport, which is what we were just looking at. It actually has broader importance. So again, carbon is fixed by photosynthesis in leaves, and I'm going to start referring to those as source leaves because the carbohydrates, they're building blocks, but they're also the currency of energy within the plant. So that leaf is the source of energy. And some of that energy, some of the carbohydrates are moved down out of the leaf to the rest of the plant, the parts that are not photosynthetic. And you can think of those as sinks, energy sinks. The stem, roots, and even new leaves as they develop before they become photosynthetic and self-sustaining. So if those tissues are dependent on transport, obviously transport is going to be very important for overall growth. And so it's going to be important for things like cellulosic biomass and bioproduct yield. And I want to start to talk a little bit about the mechanisms of transport and what we believe, what plant biologists have believed for almost that carbohydrates, the sugars, actually drive transport. So how does that work? Well, let's start by taking a big picture look at the plant vascular system, which I have here on the left in cartoon form. Very simplistically, water nutrients move up through the xylem from the roots up to the stem and then to the leaves, whereas carbohydrates move from the leaves where the photosynthesis happens, where they're made, down through the phloem to the stems and roots that need those resources. And I have sort of a blow-up view of that pipe system on the right, xylem, phloem. And what this transport is thought to be dependent on, what Ernst Munch suggested as early as 1930, is that the sugars that are made in those leaf cells have to be transported across membranes into cells linked to the phloem. And that drives the transport. That creates a pressure, which I'll talk about in a second, that moves sap basically down through a pipeline to those sink tissues, to the roots and stems that need the resources. So blown up a little bit more, a little bit simpler. And by the way, most plants, there's some variation on this in different species, but most plants transport sucrose. So we can think simplistically, sucrose is made in the photosynthetic cells. It needs to be transported. These little purple barrels are transporting sucrose into companion cells, which are linked to sieve elements, and those sieve elements make up the pipeline, the tube. And what happens, which you can kind of get a sense of here, is when there's lots of sucrose inside of the cell, water likes to follow. Water likes to move across the membrane to reach equilibrium. And when it does that, when you get more water in the same volume, that increases the pressure. And that is what is thought to drive the movement of the phloem sap, the liquid within the phloem, down. So the obvious thing to look at, if we're looking at sucrose transport across a membrane, is something that transport sucrose, like a sucrose transporter. And there are actually genes that are sucrose transporters, and they're abbreviated SUT, S-U-T. If we take one of the genetic models, this is coming from agriculture, lots of history, Zea mays, which you know as corn or maize. Zea mays actually has 7 sucrose transporter genes in its genome. It's common to have that kind of redundancy in genes in a genome, and sometimes it's actual redundancy. Sometimes those genes may have different functions. For example, a gene may be expressed at a different time in a different tissue or a different cell that gives it a different role within the plant. Or in the case of a transporter like this, it could have a different orientation within the membrane, potentially. So, as I just alluded to, it depends where these genes are expressed. So a colleague of mine, David Braun at the University of Missouri, has started doing some studies to see where are these genes expressed. And I'm going to tell you about the first one. So how do we do that? There are lots of ways, but one way, let's take one of these sucrose transporters from the membrane and blow it up. It's basically actually looks sort of like a barrel, and it forms a pore going through the cell membrane, that lipid bilayer of the cell membrane. Now, if we can pull that apart again, even further, you'll see that this forms a chain that loops in and out of the cell membrane. And so we could potentially attach something to that chain. We could attach a molecular probe, say something that's fluorescent, that we could see within the cell when this protein is expressed. And my colleague David Braun did this. He made a fusion of a sucrose transporter gene and a molecular probe, a fluorescent protein. And the beauty of this is, so he puts this into the maize genome, the gene, we don't see the gene, but when it's expressed, when the protein is made, we see that. So we can really see where this is functional, where these genes become functional proteins within the plant. So this is showing just that. This is a cross-section of a maize leaf. The arrows kind of indicate where those veins would match up to. And so the green fluorescence is showing us where the sucrose transporters are expressed for this particular sucrose transporter. This one happens to be SUT1. They're creatively named SUT1 through SUT7. So I'm going to get on the first try something that looks interesting because it's in those veins, especially the minor veins, the small veins, where we think sugars are going to be loaded into the phloem. And then those minor veins are going to feed into the larger veins like a stream into a river. So the next step, David isolated a mutant, a knockout mutant of that gene. So these plants are essentially the same as a normal wild-type plant, except for that sucrose transporter 1 gene is not expressed at all. So in these plants, we see some of the things that we would expect for a plant that can't load sugars into a phloem. We see reduced growth, and you're starting to see that even here in the young stage. We see reduced leaf number, because those new leaves are dependent on carbohydrates from other places. The plants are sterile. They don't produce either pollen or an ear. And there's reduced photosynthesis, which I show here as carbon-11 fixation. And that reduced photosynthesis is because the leaves accumulate a lot of carbohydrates. This is also what you would expect for that can't export carbohydrates. So it's making carbohydrates, it's building them up, and as it builds up, you start getting feedback to the photosynthetic systems. We've got enough carbohydrates, slow down a little bit. So now I can take that tool, that mutant, bring it to Brookhaven and use it for studies with carbon-11 to study transport. That mutant is a tool for me. So this is going to be, what I'm going to show next involves using that leaf cuvette, passing the carbon-11 carbon dioxide over the leaf, does its thing and makes metabolites. And after a period of time, there's going to be transport some of those metabolites, mainly sucrose, down through the leaf. And we can measure the speed of that movement. Again, remember, we can detectors, shielded detectors at various points and measure how long does it take to cover that fixed distance. We can also take that leaf and make an autoradiograph. This is not a photograph. This is actually showing the distribution of radioactivity in leaves of a wild type and a sucrose transporter mutant plant. So what did we find? Export from this region where the CO2 was applied, export was severely reduced in the mutant plants. And you can actually see this in the autoradiograph. This darker area here, all that darkness is radioactive sugars coming from this region that are moving down out of the leaf through the system. We see much less of that for the mutant. So that's not too surprising based on the phenotypes that we saw. What was surprising is that the transport speed, the speed of sap flow down through the leaf, how fast is the activity moving? It was decreased in the mutant, but not nearly as drastically as total export. So this was really cool. It was really kind of confounding and the sort of thing that I had to go back and say, am I doing this right? Because remember, good old Munch's hypothesis, we have to load sugars into the phloem to drive transport. So was the hypothesis wrong? And a big question remains, what's maintaining this high of a transport speed? And this could be, it's lower than this particular wild type, but that could be a normal plant. What's maintaining that sap flow? And I think what's exciting about this is I think after following this up, we're going to end up revising the plant biology textbooks, our understanding of how phloem transport works. All right, so I've talked a bit about transport, but transport is not happening in a vacuum. There's also metabolism happening. So I want to focus on that a little bit now because we need to understand both in order to understand either. So when a leaf fixes carbon dioxide, let's say it's labeled carbon-11 carbon dioxide, a lot of that carbon-11 is incorporated into sugars, the glucose, the fructose, and the sucrose. And actually, a lot of it is incorporated into starch as well. And starch is just a polymer of glucose subunits. It forms chains and branches and lots of glucose stuck together. And this is not intended to be a detailed outline of what's happening with carbohydrate metabolism. This is very simplistic. But just to get the point across that there's interconversion here. These aren't just stable. We can't just extract sucrose and say there's X amount of sucrose concentration, so that's what the plant is doing. No, because that concentration is dependent on what has been happening in the leaf over the past few hours, maybe even over the last day. So that doesn't tell us what's happening right now, because that sucrose can be interconverted with glucose and fructose. Sucrose is just a disaccharide, a glucose and a fructose unit linked together. As I mentioned, starch is a polymer of glucose, so you can get movement back and forth between those pools and also to other pools, and actually interconversion between glucose and fructose as well. So what we need is, when we look at those biochemical pools, what that concentration is dependent on is the input flux of carbon into that pool and the output flux, how much is being converted into other stuff, transported out of the tissue, and so forth. So when we measure the natural biochemical pool, the carbon-12 biochemical pool concentration, that's the puzzle. Now, if we can extract these sugars and measure how much carbon-11 that we just administered was incorporated into that biochemical pool in a short period, we get a sense of the input flux into that pool. And if you have two of the pieces, you can kind of infer what's happening with outflux in a lot of cases. So this carbon-11, carbon-12 isotopic signature could be a really important tool. So one of the first things that I did when I came to Brookhaven was I started working on a method to do that type of analysis, to measure both carbon-11 and carbon-12 sugar and starch pools. For those of you who are familiar with chemical analysis, this involves thin layer chromatography and autoradiography. For those of you who are not, basically I'm taking a coated plate, applying sample, and then separating, using solvent, running up the plate to separate compounds. And so using this technique, I'm able to separate sucrose, glucose, fructose. With autoradiography, I can image the plate. It's just a 2D image of the plate, and I can calculate how much radioactivity is incorporated into each of those sugars. I can do the same thing for starch after I degrade the starch. And then I can do a heat treatment, and under UV light, I can image the fluorescent signal and calculate the 12C concentration as well. So take home from this is I have a tool now to look at carbohydrate metabolism, not only in isolation, but in the context of the whole plant, in the context of transport dynamics, as I just talked about in the previous slides. So what did I find? Going back to this sucrose transporter knockout mutant, the sugars looked all exactly the same pattern, different actual absolute values, but the same pattern. So I'm just going to show sucrose to give a sense of what's happening with soluble sugars, and starch was a little bit different. So I have carbon-12 concentration on the top, carbon-11 on the bottom. As I alluded to before, massive carbohydrate accumulation, especially the soluble sugars. Starch also increased. But what did we see for carbon-11? For sucrose, for the sugars, we saw an increase in carbon-11, that input flux. But for starch, we saw something different. There was actually maybe even a slight decrease in input flux into starch. So what does this mean in biological terms? I'm going to go back to this biochemical pool ball and arrow model. It looks like there was an increase in biosynthesis of sucrose that contributed to the increase in that carbon-12 sucrose pool. Whereas for starch, there was a larger starch pool but less biosynthesis. So it has to be that there was a lower turnover of the starch. And you can think of this as somebody crimping a garden hose with the water on. What's going to happen above where you crimp the garden hose? It's going to start to expand. So this is the same concept. So soluble sugar biosynthesis and starch breakdown, starch turnover processes were responsive to the reduced export in these mutant leaves, the sucrose transporter mutant. Which brings up another point, because I'm analyzing this in a vacuum. What about transport? Export from these leaves, we know from the data I already showed, is decreased. So that should increase that sugar pool even more. And in fact, that's what we saw with the carbon-12 signature. The percentage increase in soluble sugars was higher, much higher than the percentage increase in the carbon-12 starch pool. So let's bring this back to bioenergy for a second, because I was telling you sugars are good. We can convert sugars into energy. Yes, this is true. This particular mutant isn't what we want, though. It's going to be small, low biomass, and we're going to lose in biomass what we would gain in efficiency of conversion. And so what I've been talking about the last few minutes is focused entirely on those source tissues, the photosynthetic cells, the leaves. What I want to talk about next is if we focus now on sink tissues, can we accumulate sugars in another part of the plant without sacrificing biomass? And that's going to bring me to the final part of the talk. This is kind of a future direction. This was a project that was funded starting about a year ago by the USDA DOE. The project is looking at sugar accumulation in sweet sorghum stems. So sweet sorghum, as the name implies, has high abundance of sugars in the stem. It's kind of like sugar cane, in that you can take the stem, this is just a stem of sorghum stripped of its leaves, you can press that stem, get the syrup, and ferment it directly without any of the pre-processing that you would need for cellulose. So that's a good thing. Turns out sweet sorghum tends to be a high biomass plant as well. That's another good thing. Unlike sugarcane, sorghum tends to have lower nutrient and water requirements. That's a good thing for growing them on marginal lands. They're drought tolerant. They have this trait called stay green, which is exactly what it sounds like. If the plant dries out, it doesn't just wilt, it actually will stay green. And if you rehydrate it, it will continue to grow. Sorghum is also adaptable to temperate climates. So those two you won't get with sugarcane. Most of the U.S. is temperate, so this is exactly what we need. It's easier to harvest, and there are lots of genetic resources. Sorghum originates from North Africa. It's been cultivated for hundreds of years. There's lots of different genetic strains with different types of characteristics that we can draw on. It's grown across Africa, Asia. India is trying to use this for bioenergy, and it's been grown in the U.S. for quite a long time as well. So we have genetic resources specific to the US. And in fact, the only reason why you probably don't know sorghum, because I'm guessing most of you don't, I didn't before I started reading this, is we don't see it in the grocery store. Some other cultures will actually use this for their own food. For us, we happen to feed it to our livestock. So what do we know about sugar accumulation in sweet sorghum? There have been a number of studies, just like there have been a number of studies in sugarcane, and it's kind of a perplexing system when you start studying enzymes, enzyme activity, what's happening inside of these stems. It doesn't make sense with what we would expect. The authors of this particular study concluded none of the enzymes under study proves to be responsible for the extent of sucrose storage in the stem. And they suggest looking at transport processes. This is a perfect project for us to do at Brookhaven. So as I mentioned, we recently were awarded a competitive grant from the Plant Feedstock Genomics for Bioenergy program. From that, we were able to hire a new postdoc, Abhijit Karve, and I want to acknowledge him. Some of his work is presented in this part of the talk. So how are we approaching this? We need a model. We need something that accumulates sugar and something that doesn't accumulate sugar, and we want it to be as similar as possible so that we can know if we see a difference, it may have something to do with sugar accumulation. Because what we want to do is understand the mechanism. If we can understand the mechanism, maybe we can manipulate it. Maybe we can breed for it. So what we came up with, myself and collaborators, was a comparison of a grain sorghum line, which is not a sugar accumulator, with a sweet dwarf sorghum line. And the reason for the dwarf is because the grain sorghum went through this whole green revolution process. It's a short plant. The sweet sorghum varieties tend to be taller. So if we want to compare like to like, we need something that's about the same size. It turns out one of my collaborators has a genetics population. He has some sweet sorghum lines that are close to the same height and about the same biomass as some of these grain sorghum lines. So the first assumption is sugars are coming from photosynthesis. So photosynthesis must have something to do with this. Photosynthesis or export of the photosynthetic product, the carbohydrates from the leaf. It turns out that's not the case. Comparing between grain and the sweet dwarf sorghum, we see no difference in photosynthesis. We see no difference in export of photosynthate of the sugars from the leaf. So what's going on? Here's where the PET imaging comes in handy. We can take those plants, start to stick them into the PET camera and see where is radioactivity, where are the sugars distributing throughout a plant after you feed labeled carbon dioxide. And it turns out for the grain, there's a lot of carbon-11, there's a lot of sugar that moves up towards the top of the plant, the apex. This is where the seed is produced, the seed head. Whereas for sweet sorghum, not so much. And we can see that when we quantify the data. If we look down at the bottom of the stem, we see a higher proportion of that carbon is going to the lower stem in the sweet sorghum than what we see for the grain sorghum. All right, so what sort of model can we develop so far? We're not seeing differences in photosynthesis or export from leaves. That must be, it's important. Sugar accumulation is dependent on that in the sense that it's dependent on sugar production, but that's not what's making one plant a sugar accumulator and the other a non-accumulator. So once the sugars are exported from the leaf and they get into the stem, there must be a difference in how they're distributed within the plant. For example, in sorghum, we saw a lot higher distribution of carbon, of sugars up towards the top of the plant, the apex, whereas in the sweet sorghum, more went down to the stem, which fits if you accumulate sugar in the stem, you need the resources to do that. And from this, I'm starting to develop a simpler model, a simple model of how we can look at this. And it's a sink competition model. Remember, sinks are the non-photosynthetic tissues, the stem, the roots, and the young leaves, and also seed head in the case of a reproductive plant. So once sugars are exported from a leaf, how are they distributed within the stem? What is the control over that? How is it regulated? It could be that there's less demand by other sinks, the apex, the roots, and so by default, the carbohydrates just kind of stick around, hang out in the stem. It's possible, but I think also there is going to be some mechanism, some process within the stem that makes it a stronger sink. It takes those sugars in, holds on to them. So since we're talking about the transport of sucrose in particular, again, the sucrose transporters come to the front. And my colleague David Braun at the University of Missouri is working to isolate sucrose transporter mutants in sorghum. This is going to be a large project to characterize all of the sucrose transporters. So that's one targeted approach. Something else, this is one of the exciting things about what we do. Every time we do something different, nobody's ever done it before, so we're always seeing stuff that we didn't expect. And it often starts out with, oh man, how am I going to deal with that? But then it turns into, hey, what's going on? Carbon-11 sugars are accumulating in these nodes. The node is actually where the leaf attaches to the stem. So this raises the thought, if sugars are accumulating there, if this isn't just a straight-through pipeline, maybe these are acting as control centers, determining where sugars go. Are they going to go up, down, or stay? And then maybe there are other mechanisms that we haven't even thought of. And another aspect of this project is we have Ishmael Dwicat, who is a geneticist at the University of Nebraska, doing more of a screening approach, genetic mapping. So we have kind of a top-down, bottom-up combination of approaches, some targeted and some non-targeted. We're probably going to find out how this works, how the sugar accumulation works, and that's going to be great for bioenergy. So in summary, if you take home nothing else, you should take home that transport is important for plant quality and yield, especially traits that are relevant to bioenergy. We've started to determine roles for specific sucrose transporters. For example, sucrose transporter 1, SUT1, is very important for export of carbohydrates from leaves. I also mentioned there are 7 sucrose transporters in the maize genome. I think in Arabidopsis there are 9. What do those other sucrose transporter genes do? Is one of them involved in, say, sugar uptake into stem cells? And so the sugar accumulation that we're interested in stems. And from what we've done so far, it looks like sink import of sugars into sink cells is important in driving that sugar accumulation in plants like sorghum and like sugarcane. And I feel, and I hope I've convinced you, that there's a key role for the PET radiotracer, the PET imaging, and delving in and elucidating these secrets of the plant and how we can use them to our advantage. So that's the end of my talk, and I'd like to thank, this work is not something that I've done on my own. This is dependent upon a team, and I'd especially like to thank the entire PET group at Brookhaven for all their support. The Goldhaber Fellowship was really important in giving me the freedom to develop new lines of research that I've presented here and to make connections at conferences and so forth. So I'm very appreciative of that fellowship. I also need to thank Department of Energy, Biological Environmental Research for supporting our group. And if anybody has any questions, I will attempt to answer them. Thank you.